Assalamualaikum warahmatullahi wabarakatuh, and hi, my name is Masturina binti Abdullah from AS2575C. So today I will be presenting my final year project proposal entitled The Efficacy of Fungal Endophytes Isolated from Menthopiperita as Antibacterial Agent. For the background study is about the increasing of pathogenic bacteria that continues to pose a serious threat and cause concern to the public. This is because pathogenic bacteria can cause a virus disease as they have capable of infecting humans, animals, and plants. They can produce virulence vector by colonizing the host tissue and going deeper into the host tissue to spread the infection. The chosen plant in this study, which is Menthopiperita, is believed to be able to fight this bacteria because it has compounds that can act as antimicrobial agents. Menthopiperita, best known as peppermint, is a hybrid plant from family Lamiaceae that cross between watermint and spearmint. It is abundantly found in Europe, North America, and Asia. It has many uses and benefits, such as antibacterial properties which can inhibit the growth of various pathogens, including antibiotic-resistant strain like MRSA. Second, it can reduce inflammation and can be used in treatment for conditions like arthritis. The antimicrobial activity of this plant involved in several mechanisms, such as cell membrane destruction, inhibition of bacterial enzyme, and biofilm inhibition. Next is endophytic fungi. Endophytic fungi is a microorganism that live inside the plant tissue without causing harm to the host tissue. Instead, it provides a benefit to the host tissue by enhancing the production of bioactive compound, providing strong antimicrobial and antioxidant activities, enhancing resistance to the pathogen by providing secondary metabolites, and promote plant growth by enhancing the nutrient uptake and producing growth hormones. The rise of multi-drug resistant bacteria that become a global threat, and even conventional antibiotics are no longer effective in curing the diseases. This is because some bacteria can develop biofilm as their protection and survival against a harsh environment, making them more resistant to antibiotics. In order to kill this bacteria, it requires a sufficient amount of antibacterial compound. However, Menthopiperita extract can only produce a small amount of bioactive compound, so the existence of fungal endophytes in the plant can enhance the production of bioactive compound. There are two questions arise from this study, which is how efficient is endophytic fungi extract isolated from Menthopiperita as antibacterial agent, and second, what are the concentration of endophytic fungi extract isolated from Menthopiperita to inhibit the pathogenic bacterial growth. The objective of this study is to determine the antibacterial activity of endophytic fungi isolated from Menthopiperita against pathogenic bacteria. Second is to evaluate the efficacy of fungal endophyte extract obtained from Menthopiperita as antibacterial agent. The methodology, the first one is endophytic fungus culture and maintenance. The fungal isolate will be cultured on PDA and will be incubated at 30 degrees Celsius for 7 days. The isolate will be subcultured on a sterile fresh medium once a month to maintain its viability. Second, test microorganism and culture maintenance. Gram-positive including Staphylococcus aureus, Bacillus subtilis, Bacillus cereus, Streptococcus mutans, and MRSA strain, as well as gram-negative bacteria such as E. coli, Pseudomonas aeruginosa, Klebsiella pneumoniae, and Salmonella typhi will be used as test bacteria. The bacterial culture will be cultivated on nutrient agar and incubated at 37 degrees Celsius. A few single colony will be selected to prepare the bacterial inoculum. Next, agar plug diffusion assay. The isolated fungi will be cut into agar plug using a cork borer. Then place the agar plug onto MHA that has been inoculated with the test bacteria. It will be placed at 4 degrees Celsius overnight to allow diffusion of bioactive compound. The plate is then will be incubated at 37 degrees Celsius for another 24 hours. The inhibition zone occurring around endophyte agar plug will be measured. Extraction and extraction of endophytic fungi. Two agar plug will be placed into aluminum flask that contains spark medium. The culture will then incubated for about 16 days in the dark environment. Fungal biomass and fermented growth from the mixture will be separated using filter paper. Using a rotary evaporator, the upper organic layer will be collected and concentrated. The mixture will be placed in the fume hood until it is completely dry. The acetate fungal growth paste will be obtained. Next is diffusion susceptible acid. Fungal growth extract will be dissolved in the acetate. Antibiotic disc will be impregnated with 20 microliters of the extract. The test bacteria will be inoculated onto MHA agar. The disc will be allowed to air dry and will be placed on MHA agar. The endophyte agar plug will be incubated for 24 hours at 37 degrees Celsius and inhibition zone developed around them will be measured. Next, MIC-MBC determination. The minimum inhibitory concentration of the fungal crude extract will be determined by broth microdilution assay using 96 well. The fungal extract will be prepared for single fold dilution. 100 microliters of extract and 100 microliters of test bacteria will be added into each well. After 24 hours incubation, the lowest concentration of crude extract that was able to prevent the visible growth of test bacteria will be recorded as MIC value. The minimum bactericidal concentration of fungal crude extract will be determined based on the value of MIC. After 24 hours, the viable cell from wells that show no microbial growth will be counted using conventional viable plate count on MHA. The MPC will be the lowest concentration of the crude fungal extract that will prevent the growth of organism. Next is time case study. A culture with bacterial inoculum and 1% DMSO in MHP will be served as growth control, while chloramphenicol will be served as antibiotic control. The culture will be shaken in a rotary elevator shaker for 48 hours. Every 4 hours, the sample will be collected for viable cell count. Only plate with the number of colonies from 30 to 300 will be used to calculate the colony forming unit. For every extract concentration and control, a time curve will be plotted. Lastly, statistical analysis. Each experiment will be conducted in triplicate. The mean standard deviation of the experiment data will be shown for each trial. So one-way ANOVA will be used to identify the differences in the means. This study will bring more insight into Mentha potential medicinal benefits, especially regarding its antimicrobial properties. Mentha, a natural remedy, can be an alternative drug against infectious disease that provides fewer side effects. Additionally, the discovery of chemical composition contained in the extract of Mentha, which has a various function, allow researchers to find a new antibiotics for the treatment of multi-drug-resistant bacteria. Lastly, this study emphasized the presence of fungal endophyte in enhancing the production of antibacterial compound in Mentha. This study is expected to investigate the role of endophytic fungi isolated from Mentha in producing bioactive compound that can act as antibacterial agent. The efficacy of its antimicrobial properties against various pathogens, including those multi-drug-resistant strain, could also be studied. So that's all from me. Thank you.